All right, welcome back. In this video, we're going to go over the condensation and peptide bond formation of amino acids. Amino acids are able to bond together to create long chains. The bond that links them together is called a peptide bond. When amino acids combine, they create water as a byproduct, hence the term condensation reaction. So whenever amino acids bind together, water is released. What's the point of this? Why are we learning this? Why is this important to our body? Well, our body's goal is to make proteins, so we can actually do stuff like make enzymes. Proteins come from amino acids, long chains of amino acids. So we need to actually make these long chains. So amino acids bind together to create long chains called polypeptides. So the term for a long chain of, a chain of amino acids called a polypeptide. When it's sufficiently long, when your body says, all right, this looks good, it turns into a protein. It folds and turns into a protein. And then it can actually do cellular functions like become an enzyme and do enzyme stuff. So here's what I did. I do a step-by-step of how this is going to work, this whole reaction. The first thing you need to do is you need to take your amino acids and expand them. Draw them out with all the bonds showing. The only exception is you don't need to do this for the R group. They don't matter. The R group does not participate in the condensation reaction at all. The only things that actually participate is the amino group and the carboxyl group. That's it. So here's the first amino acid. Here's a little test. What amino acid is this without cheating? This is aspartic acid. The protonated form. So notice that we have our carboxylic acid here. All I did is just expand it. I drew out the double bond, part of the carboxylic acid and the hydroxyl group. The amino group, the NH2, all I did is expand it, so I showed the hydrogens. That's bonded to the nitrogen. That's it. Here's the second amino acid, serine. All I did is the same thing, is I expanded it out. I have the nitrogens, the nitrogen with the hydrogens attached. So here's the NH2, I just expanded it, so I show the bonds. And it's the same with the carboxylic acid. So you're probably like, wait a second. I see a carboxylic acid. Shouldn't it just be a carboxyl group without the H at the end? When you do these amino acids and when you actually bond them together, they need to be protonated. The carboxyl group needs to be protonated. So we did all these videos showing, this formation. Let's do it in white. So all the videos I did this, right? Okay. The unprotonated form. Well, when you actually do and write out these reactions, these condensation reactions, you need to draw the hydrogen from for the carboxyl group, and you need to erase one of the hydrogens from the amino group. So it's an NH2. This is the only way this works. That's it. So on tests and exams, make sure you write it like how I'm writing it, as an NH2 and a COOH. Okay, that's the first step. Now, the second step is when you write out these amino acids, you need to make sure that the nitrogen-containing group, the amino group, is on the left-hand side. So here's what I did. Here's the amino acid. The nitrogen is on the left-hand side, right? Nitrogen's on the left-hand side of the amino acid. This is called the N-terminus. The N-terminus. The other side is called the C-terminus. C-terminus. So you write out all these amino acids from N terminus to C terminus. It is essential you do this. It is wrong if you were to give on a test, write out this amino acid and you wrote it with C terminus on the left-hand side, it would be wrong. It has to be written as the N terminus on the left-hand side. So the amino group is always on the left-hand side. So here's what you're going to do. Is the first amino acid you have to deal with, you're going to circle the hydroxyl group that's part of the carboxylic acid. So this, the hydroxyl group. Make sure you're not circling anything from the alpha carbon here. The alpha carbon stays as it is. It's the hydroxyl group from the carboxylic acid that you're going to basically do all the reaction with. So you circle the hydroxyl group. The second amino acid, the one you're binding to, you're going to circle the hydrogen that's a part of the nitrogen. Not the hydrogen part of the alpha carbon. Not this lone hydrogen. No, not that one. It's the hydrogen a part of the nitrogen, the amino group. Just one of them. Just circle one of them. And basically you're going to bubble them in together. So they're going to be in one circle together, like that. So the hydroxyl and the hydrogen are going to be circled. The hydroxyl from the carboxylic acid and the hydrogen from the amino group. Now what you're going to do is you're going to erase what you just circled. Erase the contents of what's inside the circle. So you're going to erase the hydroxyl group and you're going to erase the hydrogen. And then you're going to draw a line. You're going to make a bond. You're going to make a peptide bond now. You're going to make a bond from the carbon of the carboxyl group to the nitrogen of the amino group, of the second amino acid. Nothing to do with the alpha carbon. You're not drawing a line from the alpha carbon. You're drawing a line from the carbon from the carboxyl group to the amino group. So let's just go back to this figure. So you're going to go draw a line from this carbon, not this one, this carbon to this nitrogen. And that's called a peptide bond. You just made a bond. That's called the peptide bond. Now, what you just erased doesn't just go bye-bye. What you just erased was, still a recap here, we erased the hydroxyl group and hydrogen. That's what we erased. Notice something interesting about this? You got two hydrogens in an oxygen. H2O. That's water. We literally eliminated water. So that's the byproduct. When we bind amino acids together, we create water. So we just bonded aspartic acid together with serine. We can now abbreviate it as DS. Remember those boring abbreviations that everyone hates when I went over all the amino acids? Aspartic acid is abbreviated with a D, and serine is abbreviated with an S. So we just call this DS. We don't name it as aspartic acid and serine. Because imagine this. Imagine we had an amino acid chain that's 100 amino acids long. We don't want to say 100 amino acids. That's going to be like terrible. You're going to lose track and that's going to be terrible. No. What you do is you write the abbreviations. So you just write 100 letters instead of writing 100 words. So in research papers, if they're going over like an amino acid chain, they're always going to write it as the abbreviation. So if you had, say, 100 amino acids, right, you would do D, S, A, so, right, forever, until you're done with the chain. And you always write it with the N terminus on the left-hand side. So this is the N terminus here. That's N terminus. And you write it to the C terminus. So depending how long your amino acid is, terminus, you write it from N to C. Okay? And that is it. That's how you make peptide bonds. It's that easy. So if you had, say, we had another amino acid on this side, on this left hand side. No, let's do it here. Another amino acid here. Say we had like histidine or something like that. What would you do? So here's what I do. I want to do draw it out again. So let's just say CH carbon here, hydroxyl. And I was going to do R. Okay. What are you going to do? How do you bond this together? Well, once again, we take our hydroxyl group and the hydrogen from the amino group and erase them. We get water out of it, and then that's our bonded amino acid. That's it. If you found this video helpful, please like and subscribe. Until next time, later.